Hello, everyone. Welcome back to another chapter. This is chapter 4, which is entitled Tissue, the Living Fabric. This is actually one of my favorite topics to cover in anatomy and physiology because we get to explore histology, which is the study of tissues. Now, what exactly is a tissue? Well, by definition, a tissue is a group of cells that often have a very similar structure and a similar function that can be found working together as a unit. And when we look at the human body, there are four basic tissue types. We have our nervous tissue, which, as the name kind of hints at, is going to focus on our nervous system. So we're going to find it in areas like the brain, the spinal cord, and the nerves. We have muscle tissue, which accounts for things like skeletal muscle, smooth muscle, and cardiac muscle. We have our epithelium tissue, which is best known for forming boundaries and covering surfaces, like, for instance, our skin. The outer layer, or I should say the top layer of skin, is nothing but epithelium cells neatly lined up and stacked together. And then the fourth type of tissue, which is actually the most abundant one that we have in our body, is connective tissue. And connective tissue can be found in many different aspects of our body, internally mostly, providing things like support, binding tissues together, protection. And this is where you will find a wide array of different types, anywhere from the cartilages to the areolar and mesenchyme, bone, and even blood. Yes, blood is an example of a tissue. It is, fun fact, the only fluid tissue type that we have in our body. But more about that topic a little bit later on. Now, the way the lecture is divided is that in part one, we're going to concentrate on epithelium tissue. We'll take a look at some of its unique features. We'll take a look at the functions. We'll take a look at how it's named and some examples and where we can find it within the body. And then in part two of our lecture, we're going to go ahead and take a look at the connective tissue, the nervous tissue, and the muscular tissue. All right, so let's start with our first category, which is epithelium tissue. Epithelium tissue is also called epithelium, and that is because it tends to form a covering or a sheet that covers many of our body surfaces and cavities. In fact, when we take a look at the characteristics of epithelium tissues, we're going to see that we often have groups of cells that love to make physical contact with each other to allow for a sheet format to occur. And this will allow the epithelium cell to have a covering and or a lining type of format. So like when we talk about internal and external surfaces like our skin, our different linings of the lumens of our organs, like for instance, when we take a look at our urinary bladder or our colon. But we also see that within the epithelium tissue, you can have what they call glandular epithelia, and glandular epithelia is going to focus on tissues that are able to secrete. So for instance, when we take a look at the production of sweat and the release of it through our pores, or when we take a look at our salivary glands, which produce massive amounts of saliva on a daily basis. Regardless of which form, the covering or the secretion that you're looking at, we're going to see that the main functions of our epithelium tissue, it will always focus on protection, absorption, filtration, and then of course for the glandular, we're going to have excretion, secretion. And in both scenarios, you will have some type of sensory reception that will allow for the nervous system to intervene and obviously provide an sensory system for both internal as well as external stimulus. Now, before we get to talking about some of the specific examples of epithelium tissue, I would like to talk about the 5 distinguishing characteristics. And these are items that will always be available and that will help us identify epithelium tissue whenever we have an unknown slide or an unknown tissue that we're looking for. In order for something to be classified as epithelium, the first thing you would want to look for is what they call polarity. And polarity basically means that as the cells interact, there will be a clear difference between the top and the bottom. The top layer of cells, or the apical layer, that is usually going to be exposed to what they like to call a free surface. So like for instance, when you take a look at your skin, as you run your fingers along your arm and along your skin, those cells are all exposed to the external environment that we're normally located in. Or if we, for instance, take a look at our urinary bladder, the top layer of epithelium cells is exposed to the lumen or the cavity of the urinary bladder. So that's the apical layer. And in fact, in a little bit, when we start looking at our slides, we're going to notice that there's a lot of upper clear areas that are going to show us how there is this free surface that the cells are exposed to. The bottom layer is going to be the basal layer. The basal layer is usually the one that's going to allow for the interaction with tissue types that are underneath the epithelium layer. And most of the time, that means that they're going to be interacting with things like connective tissue. The other thing you want to look for in epithelium tissue is that the cells are always trying to make physical contact. So the cells would like to physically touch, they would like to physically interact, and part of that will come through what we call cell junctions. You can, for instance, have a tight cell junction, which means that the cells are literally just kind of like super glued next to each other. Here's my little drawing of two cells together. And what we see in a tight junction is that the cell membranes will fuse together and the cells will create almost like an impermeable barrier. You can have an anchoring junction, and in an anchoring junction, what you have are cells that are still able to make physical contact, but they will have an anchor that they share in between them. And what this anchor does is it allows the cells to ever so lightly stretch and return back to the position without ripping themselves apart. So try to visualize a beating heart. As the heart is beating, the cells are expanding and returning, expanding and returning. And when this is happening, the cells do not tear apart from each other, partly because of the anchoring junction that are being utilized. And then another one I should say is called a gap junction. A gap junction is when there is a pore or larger opening that connects the cells and allows it to share cytoplasmic content. So for instance, if there's ions like sodium and potassium in one cell, we can transfer it through the cytoplasmic content from one cell to the next by using a gap junction. Either way, whether it's tight, anchoring, or gap, we see that the cells love to make physical contact, and this is required in order for that sheet format that we rely so on for protection and covering. The third characteristic is that your epithelium tissue will need to find support from connective tissue. Partly it's because the fourth characteristic says that epithelium tissue is avascular, which means it has no blood vessels that it can rely on for nutrients and waste. Instead, it will diffuse it from the connective tissue that's below it, hence the need for support, allowing for the nutrients to go up and, of course, the waste products to come down and maintain the overall health of the epithelium cells. So in a little bit, when we look at our illustrations, take a look at some of the labelings and you'll see things like the mention of a basement membrane, which will allow for a physical boundary to be indicated where the epithelium cells end and the connective tissues start. Now, even though there are no blood vessels in those epithelium cells, there are cells that allow for nervous system interaction, so they are innervated. And this is one of the reasons why if you ever had the feeling that something's crawling on your skin and you look down and you see the smallest little ant floating around, it is because of the innervation of the epithelium tissue that you're able to sense its movement. And last but not least, the fifth distinguishing characteristics of epithelium is that it's going to be highly regenerative, which means that it can reproduce. Let's go ahead and take a look at exactly how epithelium cells are classified. And in order to do that, we have to understand that the name of the tissue will tell us two things. It will indicate for us the overall shape of the individual cell, and it will also let us know how the cells are laid out. Is it one layer? Is it multiple layers? So in order to name it successfully, the epithelium cells, we're going to have to take a look at cell layers and cell shapes. Now, when it comes to the cell layers, we're going to have two options to start with. The first one is going to be dependent on the fact that it has a single layer of cells, and that is going to be referred to as simple. And anytime there's more than one layer, we're going to call that stratified. So simple indicates one layer and stratified indicates multiple. The second thing to discuss is what is the overall cell shape. Now, when you look at the individual cell in the tissue, you're going to have to decide on three options. Does the cell have more of a rectangular or circular shape? Because then we're going to call that cuboidal. Is it more elongated, like a column? So we're going to call that columnar. And if it's neither, it doesn't look like a square, it doesn't look like a column. Instead, it looks very abstract. Some people like to say it looks like a fish scale. It looks very flat. Well, that is going to be called squamous. So once again, we're going to take a look at the layers. Is it simple or stratified? And we're going to take a look at the individual shape of the cell. Is it cuboidal? Is it columnar? Or is it squamous? And what we'll do is once we know the status of those two items, we're going to combine that information to come up with the specific name for the epithelium tissue. Let me show you an example in an illustrated setting. So here is an example of what we're looking for when we say we're looking at the cell layers. You can see right here, I have one layer of cells. Now, as you're looking at the illustration, notice that this little line right here, these represent the cell membranes. And then here in purple, we have the nuclei of the individual cells. And in this yellowish type of khaki color, that is going to be its cytoplasmic content. So as you can see right here, you have one layer of cells, which would make it a simple orientation. And in the second example below it, we now see that we have 1, 2, 3, 4, 5 layers of cells stacked on top of each other, and that would make it stratified. Now, please keep in mind that for stratified, at a minimum, you would need 2 layers. So as long as you have more than one, you're going to call that classification stratified. Now, as we migrate over to the other side of the slide, we're going to see our examples of the cell shape. So let's start with the second one. Cuboidal is when it looks more like a little square or circular in format, but you'll have a very distinctive shape to it. Once again, the purple is the nucleus and the khaki format all around it, that is the cytoplasmic content of the cell. The bottom example gives us a columnar look where there is a massive amount of cytoplasmic content that extends way beyond the size of the nucleus, allowing for the cell to have more of an elongated view. And on top, we see our squamous example, where the cell is very flat. And as you can see, it doesn't really look like a square, nor does it look like a cube or a column. So let's go ahead and take a look at some examples where we're combining the layers and the cell shape to come up with specific examples of epithelium. So our first to be called simple squamous epithelium. Simple indicates that it's one layer of cells, and squamous means that the cells are all going to be very flat, scale-like in appearance. Here is an example of a drawing of it, but more importantly, let's take a look at what it looks like under the microscope. So these little ribbons of cells, the purple and the pink that you see, that is our one layer of cell. And as you can see, the little purple dots, those are indicating the nucleus. And then the pink area or the lighter purple area around it, that's the cytoplasmic content. Because the cells are making one layer, or it kind of looks like a ribbon intertwined, there is only one layer going around. We give it the simple explanation. And once again, because the cells are very flat, that is going to be the squamous. Now, all this area that you see right here that's showing up almost like an off-white, that is a free surface. Remember that in order for something to be epithelium tissue, my apology, it needs to have an exposure to the free surface on top. And then, of course, it's going to have the basement membrane at the bottom. Now, as for the location, we can find it at different parts of the body. The ones that I would like to highlight is that you can find it in the glomeruli of the kidney, as well as in the air sacs of the lungs. And in fact, the picture that we're looking at right here are that of the alveoli, which are the air sacs in the lungs, indicating the thin layers of simple squamous epithelium. How about we take a look at an example where the cells have more distinctive shape? So what would it look like if the cells were cuboidal? Here you go. So now we're finding our way into the kidneys, and we're taking a look at the kidney tubules, and we see a lot of circular formats happening. So here's one tubule, here's another one. You have quite a few of them all over the place. And what you should notice when you look at them is one, the cells are making physical contact. And if you look ever closely, you see the membrane of the cell. There you go. So you can see they're making physical contact. In some of the cells, the nuclei have been stained, giving you that darker purple color. And when you look at the cells, you notice that they're not flat, and they're definitely not columnar. So this would be your example of a cuboidal cell, where they look a little squarish, a little circular, but definitely plumper than the squamous cells. Now this is simple cuboidal epithelium, because once again, we have one layer of cells, but instead of the cells or the layer looking almost like a ribbon, like we did in our previous examples, these cells are now making a circular format and connecting to each other. So here's another tubule. Notice how it's one layer of cells that are interacting. So this would then be our example of a simple cuboidal epithelium. Once again, lots of different locations, but we're going to focus on knowing that they can be found in the kidney tubules as well as in the ovary surface. So we saw the squamous, we saw a cuboidal. So now let's see what the columnar are all about. Here we go. This is our example of simple columnar epithelium. When we go back and look at the cells, we're going to notice that they are more elongated. So there's their columnar shape. Notice how it's one layer of cells. Here's one layer of cells. And then here's a second layer of cell. But notice how they're not making contact, because here in this white area, this is a lumen. That's an opening. So it's showing you that it's two distinctive layers of cells. This is what it looks like where you were able to look at it, for instance, in the small intestines, or if you were able to look at it in the uterine tubes, or in the colon, you would find it here. Notice how in these particular cells, besides the fact that they are more elongated, they also have a massive amount of they have a higher amount of cytoplasmic content. And in case you were wondering, what are these little white circular items that you see? Well, those are good old-fashioned goblet cells. They are going to secrete mucus. You're going to see them quite a bit as we peruse through our histology study. All right, let's see. What does it tell us? It tells us that in a simple columnar epithelium, you're going to have a single layer of tall cells. Here you go. We can see that it's going to help. We're going to locate it in, let's see, we have the non-ciliated. Oh, so non-ciliated means that when we look at the top surface, we're not going to have fine little hairs that are being displayed. So that's where we find it in most of the digestive tract. And then the ciliated variety, we're going to find in the small bronchioles, so that's by the lungs, and the uterine tubes. So that's going to be the locational terms that we're going to focus on. All right, I want to show you another example. Now this one is a little bit different. This is what we call pseudostratified columnar epithelium. Pseudo indicates appearing as if. So when you look at the picture, so here's our little picture, it appears as if we have multiple layers of cells. And part of that comes from the fact that when you look at the nuclei, notice how they're not in a straight line. They're kind of all over the place, making it appear as if the cells are stacked on top of each other. However, when you look very closely, what you're going to notice is that every single cell you see here, whether it is a little cell or a tall cell, all of them connect to the same basement membrane. So that means that all of them have the origin right over here, which means that it is one layer of cell, not multiple layers of cell. So pseudo-stratified means that it's appearing as if it's stratified, but it's really just one layer of cell. Personally, I always think it looks as if you had too many cells to fit in too small of an area, so everything kind of gets scrunched together or gets squished together. One of the ways that you can often tell that you're looking at pseudo-stratified cell is because the top surface will be covered with lots and lots of cilia. These are these long extended hairs that you see floating around. With pseudostratified epithelium, even though as you can see it's labeled pseudostratified columnar epithelium, oftentimes you do not have to indicate what the cell shape is because there will be a variety of cell shapes due to the condensed area that they are placed in. Some of the most popular locations where you will find the pseudostratified ones, especially the ciliated one, will be covering orifices, so like the ones that will line the trachea, your windpipe, as well as the majority of your upper respiratory system. And the reason that that is such a popular choice is because the cilia is an excellent way that we can take trapped pathogens wrapped in mucus and kind of float them away and cause the body to expel them either by coughing or swallow them and send them down the esophagus into the stomach to be destroyed. All right, so we've had our simple squamous, had our simple cuboidal, we've had our simple columnar, and of course our pseudostratified. Now let's take a look at what happens when you have more than one layer. So let's visit our stratified examples. And let's start off with our stratified squamous epithelium. Now, quick reminder, whenever it's stratified, that means that you have multiple layers. And here we're looking at stratified squamous from the esophagus. And we can see that we have layers upon layers of cells. Now, for some of us looking at the picture going, well, how do I know it's multiple layers of cells? Take a look at what the nuclei is doing. The nuclei are often darker stained, and you can see that you have nuclei that are stacked on top of each other, especially here at the bottom layer, which indicate the fact that you have multiple layers. And because the nuclei, or I should say the cytoplasmic content is very flat, we're going to aim at the fact that they are squamous. So this is stratified squamous epithelium, best known for covering the skin surface. And as you can see here, I highlighted for you that you have what they call non-keratinized and they have keratinized. Now, keratinized indicates that the top layer of epithelium cells are going to be dead. So there's a layer of dead cells that sit on top, allowing for a very dry environment to be created. And this is what you would find on the epidermis or the top of the skin. Non-characterized means that all the layers of cells are fully alive, and this is then used to create more of a moist environment, such as what you would find in your esophagus or in the vaginal canal. Here is a picture of what keratinized stratified squamous epithelium looks like under the microscope. So this section right here, this is all epithelium. You can see the darker spots, those are the nuclei. You can see how they're connected to each other. You can see how they're stacked on top of each other. And then notice how this particular section doesn't hold as much color, and that's because these cells are dead. So these are cells that are dead, and they actually serve a very important function because they provide a protective layer and they create a dry environment so our body can also utilize it to maintain a moisture barrier. In fact, I really like this picture because it highlights it that one of the areas that you will definitely find it will be the bottom of your feet, the soles of your feet. And the reason they like to highlight that is because many of us have gotten a pedicure where we go and they start scraping the bottom of your feet and you see all these little flakes falling off. Well, that is nothing but the dead cells being scraped off. And in fact, if you've ever gone to exfoliate your skin or if you buy a body scrub and you're scrubbing it away and you feel that your skin feels a lot smoother, that's because you've gotten rid of some of the dead cells. The other example that I like to show of dead squamous cells is whenever you're dusting your place. Dust is mostly skin cells that have flaked off, and now we have to clean up after ourselves, right? So keratinized means that there is a layer of dead cells, and right there, this is what non-keratinized looks like under the microscope. Once again, you'll have layer upon layer upon layer of cells. You can see them stacked on top of each other, and it's non-keratinized, which means that every single layer of it is completely alive. So keep in mind, where it's keratinized, it will have a dead layer of cell, which will create more of a dry environment. And when it's non-keratinized, all the cells are living, which create more of a moist environment. All right, I also want to show you another one. This one is special, kind of like the pseudostratified. This one is called transitional epithelium. Transitional epithelium is taking a look at cells that are stacked on top of each other, so it is stratified. But what makes it different is that when we look at the individual cell shape, we often see a mixed structure of squamous cells, some cuboidal cell, maybe even some columnar cells. And the top layer of cells will always be slightly larger, I like to say plumper, than the rest of the cells below. And the reason for these plump cells, and truthfully, right here in this illustration, you can see, the reason for these plumper cells is because it's going to allow them to elongate, giving it a stretching quality. Best example of that can be found in the liners of the ureters and the bladder. So when you take a look at your urinary bladder, its outermost, or maybe I should say its innermost lining, the one that's exposed to the lumen, is filled with transitional epithelium. And what that allows is as urine starts to fill in the bladder, these cells start to expand and stretch, allowing for the bladder to become larger, thereby accommodating more urine. And then as you hold your urine and finally get a chance to run to the bathroom, we see that as the urine leaves, the transitional cell, the plump cells, will go back to their normal size, thereby reducing the size of the bladder. So transitional epithelium is a stratified example of epithelium where you will have the top layer larger or plump cells that will allow for a stretch mechanism to occur. All right. Now, we started our conversation with epithelium, noting that you have the ones that do the coverings, and then we have our glandular epithelium. Glandular epithelium is going to focus on providing cells that allow for a secretion. And in this case, we often like to classify it either as endocrine or exocrine. Endocrine indicates that the product is going to be hormonal, and that will mean that it will have to be secreted internally within the bloodstream, looking for what we call a target cell. Exocrine means that the secretion is usually external or into a lumen, and it will not travel into the bloodstream. An example of an exocrine secretion, for instance, is sweat. Digestive enzymes are another example. You can also have an example where you have what we call a mixed gland, which is a gland that combines endocrine and exocrine. Probably my favorite example to give of that would be the pancreas. When a lot of people think of the pancreas and I ask them for what's the secretion, the first thing that comes to mind is insulin. Insulin is an hormone that when it's secreted, it's dumped into the bloodstream and it travels to the target cells. That would make it an endocrine secretion. But the pancreas also produces lots of digestive enzymes. They travel through the pancreatic duct and they are emptied into the duodenum, the top part of the small intestine. That is an open surface. So that would be an example of an exocrine secretion. If you think of the ovaries produce estrogen, which is a hormone, goes into the bloodstream, goes to the target cell, endocrine, but they also produce oocytes, eggs, and those will travel through a duct, the oviduct, into the uterus, which is an opening. And of course, if there's no pregnancy, they will travel through the vaginal canal and be expelled out of the body. Once again, an opening, external secretions that would make it exocrine. And of course, the gentlemen have the same setup, but they have it with their testes, that they produce the endocrine secretion of testosterone, and they produce sperm as their exocrine secretion. So just keep a lookout of where the item is secreted. If it travels in the blood, it's always endocrine. And if it's traveling in a duct onto an open surface, then it's exocrine. We are going to discuss glandular secretions with more detail in actually our next chapter, which is going to focus on the integumentary system or the skin, as well as all its accessory organs. So we'll talk a lot more about those glands. So for now, I just want to leave you with an example of an exocrine gland, which is our goblet cell that we've seen throughout our exploration of the epithelium cell. As you can see, It is filled with secretory vesicles, which in this case will contain mucus that are ready to be excreted out in an open surface. So it would be an example of an exocrine gland. And as you look at your goblet cell, I just want to remind you that if you have any questions, comments, or concern, please feel free to email me at any time. And with that, the first part of chapter 4 comes to an end, and I will see you in a little bit for part 2, where we will continue our discussion with connective tissue, nervous tissue, and muscular tissue. Talk soon.